Rules for science experiments. Store all chemicals in a safe place. Wear eye protection whenever necessary. Never mix chemicals when not instructed to do so. Be careful with any heat source. Do not taste anything unless instructed to do so. Follow all instructions carefully.
Hi, I'm Stanley the scientist, and today we're going to see what you're made of, literally. We're going to look at cells. Cells are the tiny units for all living things. Cells are so small that they usually can't be seen with the naked eye. We call them microscopic, micro, because they're so small. In order to see them, we need a microscope. A microscope magnifies things so that we can see it very clearly. Here's my microscope, so I guess we're ready to see. We'll need to make a slide of some human cells here. So to find some interesting samples of human cells from our body, we can look at our hair or eyelashes or fingernails or any number of things. I'm going to take a bit of skin from my own palm by carefully scraping sideways with this sharp knife. And then we'll transfer the resulting dust carefully onto a microscope slide. There. And we'll stain it with a tiny bit of iodine and cover it carefully with a cover slip. Then we'll insert our slide into the microscope and have a look. See, there's a cell, and there's a cell. Look at all those cells, thousands of them, from one little piece of skin. Think of how many cells there must be in all the skin on my body. And those are just skin cells. There are all kinds of different cells. It makes sense. A cell is the unit of life. Okay, I can't handle this. Hi, sorry, I don't usually cry about science, but I've just been cutting on this onion for our next plant cell observation procedure. Stanley here to examine microscopic cells under our microscope. Today, we're going to look at plant cells. Plants are very different than human or animal cells. Let's take a look at a plant cell from this onion. The onion has three main parts: the root, the leaf, and the bulb. Now, this one doesn't have very many leaf cells on it, so we won't try to make a sample out of that. However, if we cut carefully into this onion's bulb, we can come up with a piece of the bulb. And if you take a knife or something sharp and peel very carefully on the outside edge, you can usually lift, there we go, a very small tissue-thin piece of the bulb that you can then transfer to a slide and stain with iodine, put a cover slip on, and put it in the microscope and observe. We'll do that here. We've prepared our slide and stained it with a little iodine, as I earlier described. Here you'll see a nice little rectangular onion bulb cell. You'll notice the cell wall around the edge of it. That's made of cellulose. Inside of the cellulose cell wall, and that being the cell membrane, is the chloroplast, and then cytoplasm and the nucleus. Here we see the nucleus clearly stained in the center of the cell. The other parts of the cell structure are not quite as clear in this particular staining. These are some other cells. You'll notice nuclei and cell walls as we go by. Plant cells. They're a lot different than human cells, which you've seen, and animal cells as well. Next, we're going to observe some very tiny organisms that can't get away and are very easy for you to observe. That's because they'll be trapped in your little puddle. I'm referring, of course, to the microorganisms in pond water. I have some pond water here, which I'll carefully put a drop of onto my slide in hopes that there'll be something in there. Next, we'll put a cover on it, being careful not to press it so we don't squish things or push too much water out around the edges, and we'll insert it into our microscope and take a look. In the world of microscopic living things or protozoans, we see this paramecium. Notice the flagella on the left side of his little body. Oh, look, and here is a euglena. Take special note of that orange-brown eye spot. He's a very flexible little creature, always on the move, contorts himself a lot, and is very hungry. And this looks like probably an amoeba. If he is an amoeba, he'll move in nondescript ways where his body seems like it doesn't have a lot of structure. He's either bored today or very content. Here we're looking at a diatom. A diatom's a microscopic creature that has a hard shell, much like a seashell. When these little fellows die, they leave their shells behind. Many, many shells altogether are called diatomaceous earth. We use diatomaceous earth to filter swimming pools. Another type of human cell that is fairly easy to collect and observe is a human cheek cell. To collect such a sample, you'll need a blunt object such as this tongue depressor, and you'll need to open your mouth and scrape the soft inner lining of your cheek this way. Then we take the result and we scrape that sample carefully onto a slide, stain it with a little bit of iodine, and carefully cover it with a clean cover slip. We put the resulting slide into our microscope, take some time to focus. You'll notice as we pan through the world of our little slide here, we see some good definition of individual cell parts. In this group of individual cells in the center of our screen right now, you'll see tiny dots that are nuclei. Also notice the individual cell membranes, slightly thicker areas around the edge of the cells. There are some more with their nuclei. Human cheek cells, a good thing for you to observe to learn more about cells and your body. One way that microscopes are used is in the medical field. Have you ever had to give blood at the doctor's office and then you wait for the results from the lab? Well, in the lab, medical professionals are examining your blood under the microscope. To know what they're looking for takes years of training, but one thing we know they see is cells, red blood cells to be exact, and so will you if you do this experiment at home. Make sure to have permission before doing it. The first step is to put alcohol on a cotton ball like this, and then clean up your finger thoroughly and clean up a new needle. Then we're going to want to prick our finger like this with that needle and get a little blood on the surface and transfer that blood to a slide. You may use another slide to spread the blood around without polluting it a bit till we have a nice thin layer of blood. Now we want to add some ink stain to that blood and then allow that to dry for at least a minute. Here's a slide that's already dry. To that, we're going to add now a few drops of water to rinse things off, and we'll let that dry off. Here's one that's been stained and rinsed and dried. We'll give it just a little more water, a few more drops, to make sure it's clean. Then we'll let that dry a little and look at it under the microscope. Here you'll notice blood cells from my finger. There's a little movement going on here because the dye is evaporating. The water is still evaporating, so it's pulling these blood cells toward the side of the slide. You'll notice nice little round and some irregular shaped blood cells with a very well-defined edge. There they are, human blood cells. Today, we're going to study one of my favorites, flowers. Most of them smell so pretty. Others just look pretty. These mums look pretty. They don't smell particularly good. Today, we'll look through a dissection scope. It's not a microscope, kind of a field dissection scope, more like a very powerful magnifying glass. And we'll try to identify the parts of several different types of flowers for you. To start with, let's just take a look down here at one of these flowers. Now, here we've cut open a flower that's a member of the lily family. You notice on the left side of your screen is half of the flower, and over here on the right is the other half. Let's take a close look at these parts. On the female side, these are pistils, P-I-S-T-I-L, and here is a stigma on the top of a pistil, and that is on a long shaft called a style. Now, the male side is called the stamen. This has an anther where the pollen is produced, and it is on a long shaft called a filament. Now, the female side runs down into the ovary, into this area that we've cut away. The ovary will become a seed or seeds. So the pollen, which is produced on the male side, on the stamen side here, is picked up by the hair of an insect such as a bee or a small insect. Sometimes it's borne on the wind or falls by gravity down onto the female side, the pistil, and the stigma is sticky. It picks up the pollen, which runs down through this style and into the ovary and fertilizes the ovary with that pollen. Now let's go take a close look at some pollen. Here we are at a very high magnification looking at actual pollen grains from a flower that we just collected from outside. Look at the structure of each pollen grain. No wonder they make you sneeze. As you're studying flowers, you'll want to keep a reference, such as an encyclopedia article on flowers in front of you, so you can accurately identify the parts. Because God made such a great variety of flowers in the world, sometimes it's a little bit difficult to tell what the parts are that you're looking at. We've seen quite a few today. Have fun as you study flowers. Hi. Today, we're going to take a look at seeds. A seed is really an embryo inside of a seed coat. To get a better look at the embryo, we're going to cut one seed in half and then observe another seed. These will be a lima bean and a piece of corn, which I have soaked in water overnight. I'll use my knife and my tweezers to carefully open the lima bean so that we can observe the inside parts. Now you'll notice here in this lima bean that we've cut in half, most of the area around the tip of my finger is food. That little appendage or little spot down on the end is the embryo. There's the embryo right there in the lower part of the picture. You'll notice the corn seed, most of the area around the center is food. The embryo is through the middle and protruding or sticking out of the bottom there. So you see the embryo in the middle of the corn seed and food all around. Just think, if we were to plant these seeds, we could grow a whole stalk full of corn and a plant full of lima beans. Hi, today we're going to take a close-up look at mold. Now, you've probably seen mold, but do you know what it is? Mold is a type of fungus. Fungus is a type of plant. In an attempt to see the growth of mold, I'm going to put a little water, well, I have put water on several pieces of bread here and put them into jars. I've taken some of them out so you can see them. You can do this experiment at home and check it every day. Here's some mold that's a few days, about 11 days old. There's some mold here that's about 2 weeks old. And then I have some mold in the jar here that is about 3 weeks old. We'll take a look at some of this very well-developed mold and examine it under a magnifying glass or a microscope. We have a microscope here. I'll try to take some of this very carefully, transfer it onto a glass slide, and add a drop of water to it, and then, of course, cover it with a little slip cover. And then we'll put that under our microscope and observe this 3-week-old mold. Under our microscope, we can see the stem-like hyphae of this mold fungus. These tiny threads are usually made up of many, many cells. Each mold fungus hypha grows into and through more and more bread until the mold has used up most of the carbon compounds or food that it comes in contact with. When food in its area begins to run out, this mold will begin to grow long hyphae with sporocarps on them. The little black balls we see here are the sporocarps. The spores they release will ensure the survival of this fungus by being airborne, waterborne, or host-borne seeds in effect. Most fungi are saprophytes. That is, they use carbon that is fixed or processed by other living things, but only after the other living things have died. This mold is an example of a saprophyte. Hi again. Today, we're going to show you how to set up a home for a small larva known as a mealworm. Now, mealworms can be obtained in many towns at a pet shop or some shop as that. Perhaps in your area you have a pet shop that would have mealworms. These little guys are very active. To set up your home, you're going to want a small jar, such as this small mason jar. A baby food jar works very well. To your baby food jar, add some dry cereal that's not sweetened, such as this dry oatmeal. To the oatmeal, you'll want to cut a chunk of potato. This keeps the moisture just right for the mealworm's environment. Let's drop that potato in there. You'll want to change the potato at least once a week so that it doesn't get rotten. Now to this little home, we'll add our mealworm. You might put the lid on it, but don't put it on tight so he can breathe. Now you'll want to take close observation of the life cycle of this little larva, and make sure as you observe, you write down the dates and times that you observe and exactly what happened, because good observation is the key to good science. Hi, Stanley the scientist, here in my laboratory. But the things that we're going to use here today, you can find right in your kitchen at home. Common things like vinegar and baking soda. You might think that the only way to change temperature in a substance is put it on the stove or in the refrigerator. But sometimes the substances themselves cause a chemical change that will change the temperature. Let's take a look at the temperature of this vinegar right now. It's about 23 degrees Celsius. What happens when we add baking soda, also of room temperature, to it, and give that a stir? Well, let's see. We'll get our thermometer worked down in here. And look there. It's not in the refrigerator, but the temperature is dropping. It's down below 20 degrees Celsius right now. Now it's 19 degrees. And now all the way down to almost 18 degrees Celsius from 23 where we started. There you have it. Common, everyday household items have demonstrated a chemical change that has taken temperature out of this mixture very quickly. Today, let's talk some about fossils. Fossils are very interesting. Many fossils on the earth were made during Noah's flood, the great flood that covered the earth with water and sediment. Now, sediment is when soft things like dirt and powders of various sorts like limestone, powdered lime, and sand get all mixed together with water and fall into a sediment, the bottom of the water. If an animal or a plant were to fall into that sediment when it's soft before it's hardened, it would make a mold, such as this seashell being imprinted into our modeling clay here that will dry and harden. We pull that out, and here we have a mold fossil. You see, if this impression were made by a soft animal, such as a worm or something like that, and the animal would decompose or disappear, then all that's left is the mold in the hardening sediment. Now, another kind of fossil is when we have a depression or a mold in hardened sediment, and then another sediment is deposited on top of that. Let's just say that this other piece of modeling clay is soft sediment that has now fallen into the original mold. Here we have a cast fossil made out of that mold. It's not the original shell, but it's an exact replica made by casting in a mold. And so this is the way many of the fossils that we find on the earth were formed. Hello. Today, let's do an experiment with something that's happening all around us. It's called erosion. Erosion is very important to the physical surface of the earth. Well, you know, various things move earth and rock around from place to place. It could be wind blowing, or perhaps in a smaller way, ice or glaciers moving the earth, or water is a very effective erosion element. And since I don't have a glacier here in the laboratory, and if I were to blow as hard as the wind, we'd have dirt all over the place, let's base our experiment on water today. We have a pan here in the lab, and also we have a board that we'll put on a slant in the pan. Here we have a mixture of rocks and soil that we'll kind of plant here very carefully on this angled plane that represents the hard surface or crust of the earth, and then the soft crust and alluvial soil over the top. And what we're going to do here is make an imaginary plane between two mountain ranges there. We have this slight depression in. I'm not going to go all the way down to bedrock or the surface of that board. Now, what do you suppose will happen when we pour water through this area? Well, let's try it and see. Right now the water's trickling down, and we notice it taking the topsoil with it first and leaving white rocks. If we pour a little harder, well, we've gone down to bedrock in some places here, but most of the places we still have just a large valley with the heaviest rocks and pebbles left. There you have it, erosion. It's moving. Now let's do a little experiment and demonstration with something we know as volume. Volume might be thought of as the amount of space something takes up, like these two marbles. We have a large marble and a small marble. Now, to find an objective way to measure the amount of volume of each, we have to have a clever scientific experiment. This is even more useful for irregular objects, such as rocks, that you couldn't measure readily mechanically. Well, let's take a look here. We'll drop this large marble into 70 milliliters of water, and we'll drop this small marble into 70 milliliters of water, and we'll see what those 70 milliliters become. On this one, our 70 milliliters has become 76 milliliters, and on this one, our 70 milliliters has become a mere 71 milliliters. There you have it, a more accurate demonstration and measurement of volume than just looking at it with your eyes. Oh, hello. Stanley here, and you're just in time. I'm about to experiment with heat, more particularly with flame or fire, with this candle, to see what kind of changes heat or flame makes. Actually, this flame is a rapid oxidation. Well, let's see. There was a little smoke there. There's a little smoke. And we see the candle is burning and wax is melting. You see that drip? There's a physical change with this amount of heat. Now let's hold this glass plate about a decimeter over the flame. We notice no change right now, no change yet. Have to be careful with it. After all, the candle's very hot. When we get the plate actually right into the flame, the edge of the flame, we notice a dramatic change here. We'll notice a change that is soot. This happens when we interrupt the flame before it has totally oxidized the wax, and we precipitate carbon out of a flame that's not totally chemically changed. So we see while a chemical change was taking place with the wax being oxidized and vaporized, now we have soot, totally black. There's both a physical and an apparent chemical change caused by the heat. Hi, here we're going to talk about a simple scientific law known as the conservation of matter. This says that matter can neither be created nor destroyed. We can determine whether or not there's been the creation or elimination of matter by weighing what we start with and what we end up with after a chemical or physical change has occurred. When you weigh something in this manner, you're in effect measuring its mass. In front of me is an equal arm balance. This will keep track of the amount of water we have here and tell us if anything's been eliminated at the end of it all. In this jar here, I'm going to start with an amount of water that I have in this beaker, and we'll just put it here into this jar and put that into our balance. Over here, we have a pre-measured approximate same weight for now of ice, and we'll put this over here, see if we balance. Actually, we're balancing pretty closely. We might take a drop or two out of the water on this side just to make sure that we don't cheat a bit. Next, we're going to have to put lids on this so we don't lose anything to evaporation. And then we're going to, oh, look at that lid, misbalance the mix. And we'll get it balanced again with another equal lid on this side. And we're going to have to just sit back and wait for probably half an hour to an hour while this ice melts and becomes liquid water. Then we'll come back and take a look at it. Well, here we are sometime later, and we notice that our ice cubes have melted. The ice cubes appeared to be bigger than that because when water freezes, it swells up a bit. But now that they're melted, we have looks like the same amount of water. And sure, our equal arm balance is telling us that the mass is still the same. So we haven't lost anything in our physical change, transition from liquid to solid or from solid to liquid in the water. We still have the same thing. Even though a physical change has taken place, we've preserved or conserved matter. The matter has not been depleted or added to. Oh, you're just in time. I just got some fan mail. Let's read it. Oh, it's not fan mail. It's another academic question. Stanley, I'm in the 5th grade, and I have a question. It's winter now, and boy, am I cold. I can't wait for spring. I love the spring, and I wish it could be spring all year round. Can you tell me why it's not spring all year round? Sincerely, Spring Forever in Baltimore, Maryland. Well, I can tell you why it's not spring all year round. It's because the Earth is tipped on its axis. You see, the Earth rotates counterclockwise and also goes around the sun in a counterclockwise direction. Because the Earth is tipped on its axis, if this were the sun, a lot closer than it is, of course, and a lot smaller for the sake of our illustration, as the Earth rolls around here, here's the United States, the tip of Texas is way up here. Because it's tipped on its axis, we're pointing the sun toward the tip of Texas. That means the sun's way down in the southern sky. This must be winter. Not much heating going on here. As the Earth goes counterclockwise around the sun and comes over to this position, now when the Earth rotates around, the sun is shining directly on the middle of the United States all day long here. This would be summer. And of course, when it rotates between summer and winter, this position is fall. Again, this position would be winter, and so this position would be spring. This happens once a year around the sun, so it can only be spring once a year.